By saying, and I could get the tone right on this, thank you very much, Rod and Raj, for taking out 50% of my slides. I spent some of the coffee break hacking, but actually, on reflection, at least pharma are all talking from the same hymn sheet. You guys were talking about the need to be quicker, what we need to do in setup, what matters. So I've got a lot less to say about that, and a bit more we can expand on the actual data. But data's been mentioned by various people up here, but the MHRA, nicely mentioned there by Dinesh, they have their views on how data should be and how it should be collected. So we've got a bit on that for you. But just start with a little bit about GSK. Again, glad I didn't go with the video route, or it would have looked like an AZ GSK clash with who's got the best video. But we've just got a bit of sort of content here. In terms of GSK's R&D spend, 2017 is the latest figures I could get. 39 billion on R&D we spent, which is over 5 billion of the UK's GDP, which is quite handy. We've got around 98,000 employees across the globe, 17,000 of those in the UK, and globally GSK have 11,000 employees just working in R&D, so we kind of like our research. Our current therapy area is quite similar to the guys who have already spoken. So we've got respiratory, we've got oncology, immunoinflammation and HIV. But then hanging on the edges, but very much hanging on tightly, we've got our discovery research, transformational medicines, and we have vaccines, and for the time being, but ending soon, we've still got consumer health. GSK are proud to be the first company that ever had an online results database accessible to everybody, not just within the industry, not just a select few, but everyone can look at our database of what we're doing and what we're trying to do. And another area that's going off on a bit of a tangent, we've just announced a collaboration with that company you see advertised in Coronation Street, GAPS, 23andMe. So we are going to use the data to basically data mine human genetics to find where we think the opportunities are for taking research forwards. So the one left piece I did leave in was around site selection, and the site selection decision that's made, if you like, at my level. So as Dinesh said, I've been a monitor for 28 years. So the site selection decision at the beginning is, let's look at the protocol. Is it primary care? Is it secondary care? And yes, we've had the acknowledgement that it's nearly all secondary care now. But then within that, is that secondary care unit running a hub and spoke model? Have they got PICs? Are they interacting with any of the site management organisations? And sometimes are we seeing primary care patients who need to be seen in secondary care? So these are all considerations about looking where the patients are before we select the sites. Obviously, we like sites to have a decent amount of experience and qualifications. If they've got previous GSK experience, that's great, like Dinesh. Or equally, we go to the networks and we say, okay, we know nothing about this therapy area in the UK. Who would you recommend we went to? So the news about the prime sites for IQVIA, we will go to the network, they'll flag it and say, okay, these are the people we think you should approach. And then within the research team at any given site, just discussing a study, in fact the one that Dr. Bansal mentioned, having big enough sites and big enough teams to make the studies work. I'd done the phase 2 research in this drug in renal with Hull and with Leeds. So of course phase 3 came along, I sent straight to Hull and Leeds and said, right, we need a blinded team and an unblinded team. And they both said, we can't do it, we haven't got enough staff. We were able to make it work in terms of the way the renal team at Bradford was able to set it up. A little bit of jiggling and juggling, a little bit of stealing nurses from elsewhere, but we got the team together. So it was a way of making the capacity work for itself. There's been a little bit of talk about the patient populations and feasibility and referrals. There's very much difference between the acute studies and the chronics. The chronics, we can do all the planning and loving it and get the patients lined up, this works. The acutes, less so. We have to sort of look at historic data and say, well, previously, we've had patients like that come through, and we all know how to cure any disease, open up a trial and the patients stop coming. Works every time. But as much as we can, we go back to existing data and databases to say, these are the patients we think we've got. So having those sorts of databases helps us out as the sponsor. Equipment and infrastructure, obviously there's trial specific, but sometimes the kit's there and we ask you to use it. Sometimes we have to bring it to you and say, right, this is what you need to do with this piece of equipment, and we'll get the manufacturers in to train you up on it. I'm glad I put especially R&D under support departments, because actually they're probably becoming the biggest support department that we need in all of our centres now, because you're the guys who make it happen at the outset of the study, and that outset has already been mentioned as being the crunch time. And one little bit, which leads nicely into what Claire's going to say, we're getting more pressure from inspectors and regulators to find out whether you guys have SOPs and procedures for the things you do routinely, and to make sure those SOPs neither conflict with ICH GCP nor with the SOPs of the sponsor companies. Because if we turn around and say, right, we absolutely love your site and we'd like you to do the study, are you ready to go? And they say, well, actually, no, if your patient in your study, if their BP drops below a certain point, we are mandated to stop study drug. And you're like, oh, okay. So that can be quite a big impact. So you'll find more and more that people coming to you with studies are turning around and saying, can we see your SOP on consent or your SOP on lab processing or whatever? And that's the pressure coming through. But the next thing, which I hope is the next slide since I've chopped it, yes, is the quality of the data. So Claire.
Hello. So this has become a bit of a trend. So we try and follow trends. So we look at the audits that the MHRA and the EMEA perform on a regular basis. And one of the trends that's coming out of that at the moment is the data quality. Obviously, data quality is really important. We need good data to prove that the drugs work. And certainly a focus at the moment is this nice little acronym called ALCOA CCA. So for all you research nurses and investigators, this is one of the possible boring elements of the SIVs. But it's a real focus at the moment, and certainly we've got some kind of inside knowledge that the MHRA are going to start looking at this in audits. And it's certainly something that we look at on a day-to-day basis when we come to sites and look at your source data. And these are our expectations and what we would hope that you would meet. I suggest it's a partnership and you are collaborating with us. We're not here to kind of audit and inspect you, that's someone else's job. The idea is that we're here to help you and give you ideas and share ideas possibly from other trusts that do it well. I know the research team do it really well here, so Karen is a good point of contact if you have any issues with monitors. But do call them out, ask them what they think good source data is and what they're looking for. And it's definitely a collaboration at the beginning. You should set up what the expectations are. I guess it's changing focus. This is slightly a paper-based analogy, but maybe now we're moving to electronic medical records, and these items are still relevant, but it's how they transpose the cells from paper into electronic formats.
So, just to finish us off, people have been very interested in what's going to be going forwards. So the GSK pipeline as it stands at the moment, within respiratory, we've got compounds in development for COPD, for hypereosinophilic syndrome, which causes severe asthma, asthma itself, acute lung injury, and pulmonary arterial hypertension. We've got an enormous space within oncology, and in fact you've probably seen in the press that we've recently linked up with Tesaro to widen our oncology. So we're looking at ovarian cancer, lung cancer, multiple myeloma, sarcoma, breast cancer, and the hematological disorders, which covers pretty much everybody in the room, I think. We've got a large research area in immunoinflammation, so that covers lupus, including down into the paediatrics. We're looking at Sjogren's syndrome, rheumatoid arthritis, ulcerative colitis, systemic sclerosis, which I've just acquired from Claire, and inflammatory bowel disease. We've got a large research area in HIV, in Hep B, and in bacterial infections. And seeing Hep B on that list makes me feel particularly old, because the first hospital study I did was in Hep B when we were trying lamivudine in 1991 to see whether it would back off Hep B after liver transplants. It hadn't even been thought of as an HIV med in those days. And then one that I particularly picked out on there, we've got renal and nephrology conditions under treatment, we've got chronic kidney disease, and the area I was working with Taryn and Jodie was around renal anemia. So we've got all sorts going on, and to add on to that, which we can't list because there's too many, again, we've got a great deal of work going in the rare diseases, what we used to call the orphan diseases. because you need to be a company of a certain size to be able to look at these ones, because they're never going to make the money back, but there's a moral imperative to try and research them. So I think, yeah, that's us.